presentation. So Dr. Dunia now is the screen clear to everyone?
Yes.
Okay. So now Dr. Dunia will give the last part for her antigen, about the antigen presentation. Dr. Dunia, the floor is yours.
Okay. So I'm going to talk now about antigen presentation. So it's very important that you understand the major or the principal molecule that are involved in antigen presentation, which is the major histocompatibility complex, MHC. And MHC is a set of cell surface molecules encoded by a large genetic complex with multiple loci. So MHC, you'll find MHC on almost all the cells in our body. And the main function of MHC is to bind or to process peptides from antigen that entered the host. And so the MHC, I always look at it as if it's like a pocket, like a bag or a pocket, and the peptide or the antigen is in the cleft or in part within this pocket. Also, MHC serves as a unique identification marker for every individual. So it is not as broad as ABO blood grouping, and it's also not as specific as fingerprint. It's kind of in between. So you will find individuals sharing with you your MHC, but the chances are not very high. Also, MHC determines the compatibility of donor and organ transplant, for organ transplant. And I think this is the interest for many of you. And so the MHC of the recipient has to be the same as the MHC of the donor. So the donor and the recipient MHC has to be identical in order for a transplant to be successful. So what about the structure? How does the MHC structure look like? And Fadi, can you please change the slide? Yes. So we have two MHC, class 1 and class 2. Class 1 consists of 4 domains. We have alpha 1, alpha 2, and alpha 3, and also beta 2 microglobulin bound to these domains. And for MHC class 2, it consists of 4 domains, alpha 1 and alpha 2, and beta 1 and beta 2. And as you can see, it is bound to the membrane of the cell, and the top of the MHC molecule has like a cleft. And in that cleft is where the peptide of the antigen will bind. So human leukocyte antigen. Human leukocyte antigen is the major histocompatibility complex in human. So you can say MHC or you can say HLA, both mean the same thing. But when we say HLA, we mean MHC in humans. So HLA complex code for the MHC protein, and they are located on the chromosome number 6. So they are around like 4,000 kilobase pairs in length. So there are many antigens in that region, and we can classify them into region 1, 2, and 3. So MHC region 1 comprise the genes that code for HLA-A, B, and C, and MHC region 2 comprise the genes that codes for HLA, DR, DP, and DQ, in addition to some non-classical genes, which are DM, DO, LMP, TAB. Then we have region number 3, but it's not, these proteins are not involved in antigen presentation, so we are not going to go into the detail of region number 3. Okay. So if you want to describe the HLA or the MHC in human, we can say it is very polygenic. So there are many genes that encode for that, for these proteins. So we have, as I said, HLA-A, B, C, DR, DP, and DQ. And we also have them from both parents. So each individual will have a pair of HLA-A, B, C, DR, DP, DQ. In addition, of course, these are the classical protein. They're also non-classical HLA, but we're not going to cover them in this lecture. Also, HLA is polymorphic. So what does that mean? So we don't have only one HLA-A and one HLA-B. No, we have many different types. So there are, today we can say there are more than 6,000 different HLA-As, 7,500 more different HLA-Bs, and so on. So, and this is not fixed, it's actually changing. So every day we are discovering new HLA alleles. So if we type an individual, this is an example we have here. We can say an individual has HLA, for example, A2, A26, B50, P51, HLA-P7. Yes? Can you please unmute your microphones? Thank you. And so as you can see here, you can have, when you type an individual, you will have the both alleles of that individual for A, B, C, DR, DP, and DQ. Next slide, please. Okay, so here is an example, just for you to understand it. So for example, let's say if the father, and we give here color code so to make it easier. Let's say the father has completely different HLA alleles from the mother. And let's assume that, for example, the blue is HLA. So this is only an example for one of the HLA classes, A type. So A could be 1 and 2. And for example, the mother is A3 and 4. Let's say that. Okay. So what will be the children? The children could either be, they would take one allele from the father and one allele from the mother. So the children can be 1 and 3, or 1 and 4, or 2 and 3, and 2 and 4. So there are only 4 combinations that the children can have. And you can see here, so the children could be completely different, or they can be 50% sharing alleles, or 25, or 0. Okay, and this is what it makes it more complicated. Next slide, please. So what about the expression? Where do we find MHC? So MHC class 1 is almost on all the cells in our body, and it is recognized by CD8, which are the cytotoxic T cells. But MHC class 1 is expressed only on antigen-presenting cells, which include dendritic cells, macrophages, and B lymphocytes. And they are recognized by the T helper cells, which are the CD4 positive T cells. So in order to have antigen presentation, we have to go through steps. So the first steps, we call it antigen capture. So Fadi, can you please change the slide? Yes. So we have, what does it mean? What does it mean to capture an antigen? To capture an antigen is actually getting the pathogen inside the cell. So there are different ways. We're not going to go into details here, but you can have phagocytosis, endocytosis. So there are different ways to get an antigen inside a cell. And then within the cell, the antigen can be freely in the cytosol, or it can be in vesicles. Next. So once it's in the cell, now we need to process it, because inside the cell, if it's a pathogen, it will be a full protein, or a full pathogen, or bacteria, or a virus, but we need to degrade it. We need to cut it into pieces. So this is called processing. So instead of having one large macromolecule, you will have small peptides. And these peptides will then bind with MHC molecules. The third stage, we call it antigen presentation. And this stage is where the degraded peptides will bind with the MHC and transported to the outside of the cell. So on the surface of the cell, we will have then the MHC bound together with the peptide of that antigen inside the cleft, as I showed you earlier. Next slide. So who are these antigen presenting cells? As I said, they are specialized cells because they have the ability to express MHC class 2, but also they can give the co-stimulatory signal to T helper cells. And they include, as I mentioned earlier, dendritic cell, macrophages, and B cells. So what determines whether this antigen or these peptides are displayed with MHC class 1 or MHC class 2? Which T lymphocyte will actually recognize the antigen? It all depends on the antigen, the type of the antigen, and the route that the antigen took to enter the cell. So we have two major classes. We call them endogenous antigen and exogenous antigen. The endogenous antigen are the one that enter the cell and enter the cytosol. Like viruses, for example. Viruses, they enter the cell and they are present in the cytosol of cells. Also tumors, we can call them endogenous antigen. But exogenous antigen are like bacteria. They don't need to go inside the cell.
I need headphone.
So what happens is we capture these bacteria and we force them inside a cell. And then they will not be in the cytosol, they will be inside vesicles. And then when they're inside vesicles, then they will be presented with MHC class 2 on the surface of that cell. So if we go back to the endogenous antigen, as I said, they are like viruses, and they are they need to be processed, but there are also different ways of processing these antigen, whether they're endogenous or exogenous. So they're endogenous proteasome. So it's like with you can where you put your steak in, and then you get like the ground beef. So you have this whole molecule, for example, the virus, and the virus enters that proteasome, and then you have small peptides of that antigen. Then they need to be pumped into the endoplasmic reticulum, because in the plasmic reticulum is also where the MHC class 1 is created. So in order for these peptides to move from the cytosol into the endoplasmic reticulum, we have transporter protein. We call them TAP1 and TAP2. They actually grab these peptides and transfer them to the lumen of the endoplasmic reticulum. Okay, so as I said, the first stage here is when you have the virus inside the cell. It enters the proteasome, and I don't know if you can see that purple thing in the middle of the slide. This is the proteasome. Then we have the peptides coming out of the proteasome and transported to the lumen of the endoplasmic reticulum. At that same site is where we have the creation of the MHC class 1 molecule. So I'm not going to go too much into the detail here in the folding of the MHC, but what I want you to understand that both the MHC molecule, MHC class 1, and the peptide will meet here, and then they will be transported through the Golgi apparatus to the surface of the cell. And as I mentioned earlier, if it's an MHC class 1, then that means it could be any cell. And it makes sense because any cell can be virally infected. And also any cell can become a tumor cell, right? So it makes sense that these MHC class 1 are presented on any cell. What about exogenous antigen? So exogenous antigen, I told you they are like bacteria. It's actually the slide before that, Fadi. Anyway. So we have the bacteria, and the bacteria will be entered into the cell by endocytosis or phagocytosis. Okay, so we have this whole macromolecule in a vesicle, and then in the vesicle, it will increase the acidity of that vesicle, so that will degrade that pathogen into peptides. At the same time, we have the MHC class 2. The MHC class 2 is sensitized in the endoplasmic reticulum. But now the MHC class 2 will not wait in the endoplasmic reticulum for the peptide. No, they will actually meet outside. So the MHC class 2 will also be carried in a vesicle, in a different vesicle. And these two vesicles will meet and fuse together. So you have the MHC in one vesicle and the peptides of the pathogen in the other vesicle. When they fuse together, now the peptide can enter the cleft of the MHC class 2, and then they will be transferred and transported to the membrane of the cell and presented on the surface. So the MHC class 2 with the peptide is a good recognition and a must for the activation of T helper cells. Next slide. Okay, so here you can see here some more details, which I'm not going to go into. There are some parts that are blocked in the MHC class 2 in the beginning, but then later on it will be free for the peptide to bind and join and then transfer to the outside of the cell. Okay, so what I want you to remember is if it is a virus, it will be in the cytosol. And it will be presented with MHC class 1 because that is important for a cytotoxic T cell to recognize it, because we know that, which is here on the right side, any cell can express MHC class 1. So if the cytotoxic T cell is activated, these cells are actually, I call them, they are like serial killers, because once they are activated and proliferate, you have this army of killer cells. But if the antigen is a bacteria, then the bacteria will be internalized in a vesicle. The vesicle will bind to MHC class 2 and transported to the surface to be seen by a CD4 or a T helper cell. A T helper cell, once it's activated, it will proliferate, and then it is ready to activate B cells. B cells are waiting for the T cell, and they already received their first signal by seeing the antigen floating around, but they still need the help from T cells. So once the T cell is ready, they will bind to B cell and activate the B cell. B cells then will become activated, they will proliferate, and then they will produce antibodies. So if we think bacteria, we think B cell, we think antibodies. And if we have viruses, we think cytotoxic T cells and direct killing. Okay, so this is the take-home message. And I think this is our last slide. Yes, thank you very much.
Thank you very much, Dr. Dunia. Well done. Anybody have question to Dr. Dunia?
Can I ask a question, Dr. Dunia?
Sure.
Thank you for a nice presentation. Could you please just explain if there is any immune response against the MHC itself? For example, if we use a tissue, can induce any immune response against the MHC, or all the MHC in all the individuals is the same? There is no any immune reactions in exchanging MHC.
So within an individual, there is no recognition of our own protein. But this is what happens in transplant. So in transplantation, if the MHC of the donor and the recipient is not 100% match, the recipient will recognize the donor MHC as foreign, and they will start attacking the MHC of the donor. And also vice versa, you can have the opposite, where the donor recognize the MHC of the recipient as foreign. Yeah.
You have interesting questions on the chat. Somebody asking how the antibodies against vaccines against viruses is created. I'm also thinking about this question.
Well, we do have, sometimes what happens is the viruses, they internalized into the normal route is into the cytosol, and this will activate, of course, our the route that I just explained to you earlier. But sometimes you could have the activation of some viruses through vesicles. And this will activate, of course, then the T helper cells, and you have antibodies. And actually, antibodies are very important for neutralization of viruses. Because we always we think like the no breaking rules, but this is not actually what happens. So we always, when we think about antibodies, okay, antibodies are only important for bacteria, but actually antibodies can be important, and it has been actually shown in many papers and research that when the antibody because of the neutralization, because of binding to the virus, and they have been shown, I think, is it measles or mumps, where they've seen that the antibody play actually a very important role against viral infection. So, but there are still a lot of things that we need to learn, and there's always something new always coming out. We learn about the main thing or the basics in immunology, and then one paper comes out and it just tells you something new and something that we have to kind of digest and see how and try to find out the mechanism and how this is working. So there's no surprises with immunology all the time.
Dr. Dunia, here is a question from someone who just talked about in relation to the polymorphism of MHC class 1 and 2. Any thoughts about immune tolerance pathways?
Sorry, what?
In relation to the polymorphism of MHC class 1 and 2, any thoughts about immune tolerance pathway?
I think this will be covered by Dr. Rabab. I think she will talk about this. So I'm not gonna steal her light.
Okay, so now thank you, Dr. Dunia, for your presentation. You did great. Now we'll have break until 5 o'clock, so probably 12 minutes more, and then we have two great talk about Dr. Rabab Al-Attas. At 5 o'clock, we're supposed to start 5 Saudi time, so like 12 minutes more. So we'll rest, have a break, have coffee, 10 minutes, 12 minutes, I'm back, please. Thank you.
Thank you. Bye-bye.
Thanks.